Welcome to Classifying and Identifying Fungi presentation as part of our Exploring Fungi unit. I'm Sandra. Today we'll be discussing the microscopic features of fungi. So this is where you need to use a microscope to actually identify fungi. Remember last time we learned about the different features of mushrooms and the stipe and the cap and how the gills are arranged and all of this cool stuff with many different words. And before that we looked at the general shapes and how we can group fungi into those. Now we're going to look at the mini stuff, like the microscopic features, using a microscope and seeing what is happening under there that we can't see with the naked eye. This allows us to really make a good identity on a fungi, put a name to it. You've got your microscope, you're looking down, and you might see something that looks like the image on the right. This is a basidium. All it is a type of structure that some fungi are grouped based on that. It basically produces little spores. So the two images you see on the right are the basidium, part of the Basidiomycetes group of fungi that produces spores on short appendages called sterigmata. So it's very complex, I know. I find it complex too. And the sterigmata projects out of a club-shaped basidia, usually containing 4 spores and rarely 2. And so when you look under the microscope and you see this sort of devilish-looking thing with little spikes sticking out and a little spore sticking out, that's when you know, this belongs to the Basidiomyces group. The macrofungi or the visible fungi in nature basically fit into these two main groups. Once you go down the microscopic way, you go, wow, okay, there's a basidium there. Now the opposite of that is the Ascomycetes group. And what these fungi have is asci, that's plural or singular, ascus. So Ascomyces produce spores in a tube-shaped container known as an ascus, usually containing 8 spores. So you can see the spores being actually in these tubes, club-shaped sort of structures, and the little spores fit in like peas in a pod. And that's when you know it belongs to the group Ascomyces. It's just a different way the two groups of fungi bear their spores, and that's how we can identify them. Isn't that cool? Just to show you what spores might look like and what they actually are, they are the reproductive structures giving rise to a new individual. Spores can have a variety of morphological forms, or basically it's saying spores can look different to one another. And so if an individual spore under the microscope looks different to another one, we know it comes from a different species. It's like one pair of humans have a baby that has specific shape of face, eye color, hair color, and another couple has a different one. So we know they're both human babies, but they come from 2 different couples. And it's kind of almost similar, except in this case, the fungi are related, but they actually form different species. So the spores that look the same can breed between each other only and not with spores from other fungi. So I hope that makes some sense to you. But if you look on the image on the left, puffballs, when they get mature, you can actually poke them and the spore just goes flying up into the air with great force. It's quite cool. Looks like smoke, but it's composed of millions and millions of tiny little spores. And these are what makes baby mushrooms when they mate. When they come together and exchange their genes, they form mushrooms or mycelium of a specific species of fungi. And you can see in the right side image that we've got spores of different shapes. And once again, scientists had to name those shapes. So if a spore is round, we have a globose morphology. If we have something that's more like elliptical, then we call them elliptical. If they're really long, we call them oblong. If they have little features, like for example, little nooks and crannies, we call them warted. And some are wrinkled. And some are spiny and have little spines, and they're all called ornamentations. So spores can actually have structures on them, and they all serve a purpose for the spore, depending on the species, So it's not just to make them look pretty, but it helps us to identify the type of fungi we're dealing with based on their spores. And this is a very fun thing to do, is to look at spores under the microscope. Just to give you some examples of what spores of the Ascomycetes might look like, and these are in the ascus. Remember the really club-shaped sort of like a pea in a pod sort of structure. So the ascospores are inside an ascus here, and the spores, individual little balls, are the spores here. And the same happens here. Look, some of them look like they're almonds. It's very cute, but they're tiny little underneath the microscope. You can just barely sometimes see them. They're tiny little things, but really cool. And you can stain them with different stains, so it makes them more visible. And so they're just different images of different ascospores, and some of them have a wall around them, as you can see, and that protects the spore and the genetic makeup. And then we have some that are round with little ornamentation on them as well. So they're also ascospores. And then we've got the basidiospores. On the basidium, remember I said the basidium has this funny looking shape, very different to the ascas. It's got these little horns sticking out, and the spore then comes up at the top called the basidiospore. So that's just to show you. And some basidia are differently shaped than others. So the little horns stick out and they're very, very long, and then the spore will be born out of those. Just to show you some images of microscopic stuff. Once again, this is just more about the basidiospore itself. You can just see the structure under the microscope. And you can imagine how tiny these are. They are magnified thousands of times in this case. And that's just to show you something that you could not see with your naked eye. So very cool stuff. Now, you can actually make your own spore print because spore prints come out in millions. Sorry, because spores come out of mushrooms and other fungi out in millions, literally. What happens is you can actually lay a mushroom upside down on a piece of paper. If it's white or black, because some spores will vary in color, of course, they all vary in color. So if you want your spore print to come out, you can put the mushroom on both types of paper, black or white, and then you'll know what color the spore is. If you put very dark spores on a dark piece of paper, obviously you won't see them very much, and this is why we use different shades of paper. But on the left hand here, you've got a mushroom, and then that mushroom was, placed upside down for quite a while, maybe a day, maybe or two, maybe overnight, covered with a piece like a glass or a cup, and left to drop its spores. And then when it drops its spores, you can see the actual pattern that the spores create. So you don't move it during that time when it's doing this, and you can see just these beautiful images which can be used as artwork. Beautiful, beautiful images come out of these fungi dropping their spores, and you can then preserve them, spray them with hairspray, and put them on your wall if you like, as a fun thing to have. And of course, scientists can use these. I'll just go back here. You can use that and actually then scrape it off and put the spores on a glass, a special glass that we use for microscopy, and you can observe them and see what shape they are and describe them and name the different features on them. So once again, this could be used in science, not just art, of course, and that's its normal purpose normally. But it just happens to be pretty. So also you can look under the microscope at the hyphae, or the thread-like cells which form the mycelium that we've been learning about throughout the unit. And the mycelium, that's a microscopic image of it on the left, but it looks like at the image on the top you can actually see with your naked eye. So the hyphae, you can see individual hyphae in the image on the bottom. And if you were to draw it, they have specific features as well, like the septum. So that's like the dividing wall. So you can base your identification of fungi on how the mycelium or the hyphae actually look under the microscope. Some have a septum, some don't, and some have things called clamp connections. So these little sort of appendages that stick out and grab around the hyphae. And so some have them, some don't, and this is once again a really lovely way to go deeper into identifying your fungi based on these features, whether they have them or not. Now, another great thing that's kind of missing in Australia a lot, we don't have many fungal keys, but what you can do once you've learned all the, or you have images and diagrams in front of you, and you kind of learn a little bit about the different features of fungi, you can go back and key them out. So, you can say, for example, with this one on the Queensland Mycological Society, you've got number 1, stipe is yellow. It yellows distinctly when scratched. So if you scratch your stipe of a particular mushroom you're holding, does it go yellow? If it does, you go to number 2. But if it doesn't, you have to keep going. And finally, you get to a level where you can actually see, if I go to number 2 because it is yellow and it's got a cap that's white, it's cream or occasionally pale, greyish brown, is yellowing when scratched, at least at the margin level, then it must be Agaricus, Xanthodermis. Okay, so does that make sense? So we go from every number we find out whether that feature exists. In this case, it's a color of the cap or the stem, and keep going down. And if it doesn't have it, you go to a different number and you keep going, you know. And this is how a key functions. It's really fun, and you can do this with your teacher when you're identifying your mushrooms that you buy from the shop. And so I encourage you to play around, and this is sort of I'm introducing you all to the science of taxonomy. So it isn't easy. That's why none of this is easy. It's fun, and it can be, but it requires a lot of persistence. And this is where, you know, people study in depth how to classify organisms. And in this case, we're studying fungi, just to show you how difficult it is, but also how fun it can be. So if you choose to go down that path in your career, that's something that needs to be explored, because there are not many fungal taxonomists in Australia. In fact, I think there's only 2 or 3 that are actually paid taxonomists. So it's a lovely area to start sort of studying and getting into, and helping the environment by understanding the different fungi and their role in ecosystems. So that's just a little introduction that we've, you know, had, and it was lovely to speak to you, and I thank you for your attention. I'm just trying to find out what my next slide is. Here we go. It's a thank you slide. If you want to learn more, you can always visit Myco-Mania on Facebook. That's my page where I post a lot of good stuff, a lot of scientific articles on there for you guys to read and enjoy. Or if you want to ask me any questions, you can email me directly at sandra@woodfordia.com. So thanks once more, and hope to see you out there collecting fungi. Enjoy, guys. Thanks. Bye.